Hi, Megan.
Hi.
What's your question?
I just wanted to know about nutrition, like what it is and stuff.
Okay. A basic definition for you to know for your Regents exam on nutrition is obtaining nutrients that will be used as building blocks to build you or as energy. Now, for example, I was saying earlier that your body is mostly protein. It's not like you can make your own protein. You have to ingest, you have to eat the building blocks, which are amino acids. So what happens with nutrition? Say you eat meat or steak or some kind of protein. It comes into your body as a big chunk of protein. It has to be broken down into the small building blocks, amino acids. Then that nutrient, amino acids, goes in your bloodstream, and say you're working out and you're building bigger biceps. The amino acids will travel to your biceps and build your muscle bigger by filling in the muscle tissue and making more protein. So nutrients build you and they give you energy. Like if you eat sugar, that goes to your mitochondria and your mitochondria make ATP for energy. That saying you are what you eat is 100% true. If you don't put it in, protein for example, you cannot build muscle. Did I answer your question?
Yes, thank you.
You're welcome. Hi, Chelsea.
Hi.
What's your question?
How does the restriction enzyme work?
Restriction enzymes are enzymes used when scientists working with DNA, for example, kind of like, this is like a crime scene kind of question. If you find blood at a crime scene, of course it contains DNA because they're cells. And I think I mentioned earlier that one strand of DNA is like 3 billion base pairs long. That's a really long piece of DNA to be analyzed. Restriction enzymes are added to a sample of DNA, and it chops up the DNA into smaller fragments. Restriction enzymes are designed to bind to the DNA and then cut it. For example, say you have a base sequence on your DNA, AATTG. The scientists would literally design an enzyme that matches that opposite base sequence, and once it attaches to the DNA, it will cut it there. So the function of restriction enzymes is to cut DNA into smaller, manageable pieces that then, did you hear of gel electrophoresis, I think, in class probably? Then you can load the DNA into what's called a gel, and you hook it up to electricity, and the electricity sorts the chunks of DNA by size. And then once you stain it, you literally get this design that you can match up one person's DNA with another. And so the restriction enzymes are used to cut up the DNA into specific pieces so it's easier to match one person to the other. Did I explain that okay for you?
Yes, thank you.
You're welcome. Hi, Riley.
Hello.
What's your question?
What is the function of a restriction enzyme?
I just answered that one. Restriction enzymes are proteins that are designed to chop up the DNA into smaller, more manageable pieces so that you can then use the DNA to analyze two different organisms' DNA and match up the chunks that the restriction enzyme has made. Restriction enzymes, they're like genetic scissors. They cut DNA into certain fragments. Okay?
Yep.
Okay? One of the hot topics that you will have to know a lot about on this Regents exam is ecology. The definition, the study of organisms and their interaction with the environment. At least a third of your Regents exam, I bet you're going to find ecology questions. Usually when we start ecology, we talk about the biosphere. If you break it down, bio means life, of course, biology, the study of life. Sphere is a sphere, circle. And so the biosphere is in reference to planet Earth. It includes all the land, because of course organisms live in the land, on the land, all the water. and the air. So anywhere an organism can live on Earth is included in the biosphere. Then we talk about living parts of the biosphere, the planet Earth, and the non-living parts, and we need both. The living parts are the biotic factors: plants, animals, fungus, bacteria, protists. Again, remember, the prefix bio means life. The abiotic factors are the non-living factors: water, soil, sunlight, energy, living space, wind, weather, pollution. And those two things, the biotic factors and the abiotic factors, interact together, kind of to help maintain a population. As a matter of fact, the abiotic factors can become what's called a limiting factor. For example, if there's not enough light for plants to grow, they'll die. So a limiting factor, a limiting abiotic factor, would be the amount of light. That could happen on the floor of a tropical rainforest, for example. Because the trees are so tightly packed together and they form a canopy, sunlight doesn't reach the floor of a tropical rainforest. The levels of organization in ecology. Now, you could be asked in the form of a bullseye diagram or a pyramid the proper order of organization of factors in the biosphere. Of course, starting with biosphere as the largest, which is then made up of many ecosystems, which is then made up of many communities, populations interacting with each other. Then a population, by definition, a population is all the organisms of one species in one area at one time. Within a population are organisms, a living thing. And finally, the habitat. Habitat is an organism's environment. It's their home. The habitat for a squirrel, for example, is an oak tree. Habitat for a polar bear is the tundra. So habitat is where an organism lives, or where a population or a species lives. Hi, Gloria.
Hi.
What's your question?
I want to know what's Gregor Mendel's theory?
Gregor Mendel? Gregor Mendel is the father of genetics. He was an Austrian monk. He studied dominance and recessiveness in genes. He figured out that genes must come in pairs and that one must be dominant over the other. He did this studying garden pea plants, actually. The reason he studied peas to determine genetics and heredity was because peas reproduce super fast. He could control their reproduction by pollinating the flowers himself. And he could get multiple generations in a very short time. Prior to him, no one was really studying heredity because you can only actually study in one lifetime maybe 3 generations of people. But with garden pea plants, he had hundreds of generations to study. And basically what he did is he theorized that if I crossed a whole bunch of tall plants with a whole bunch of short plants, he believed you wouldn't get medium height. He believed one of those traits was dominant and that's what would show up. And that's what happened. He got all tall plants. So that's Gregor Mendel, father of genetics.